so far. Number one, we looked at the elimination of CO2, and we do that through the lungs. We looked at the elimination of fecal matter for the purposes of eliminating things that flow through the digestive canals, or through the alimentary canal, and any things that we don't absorb get excreted out. But we do have to look at another mechanism that's tying in the cardiovascular system with excreting wastes, and that is going to be the urinary system. So not only is it going to be excreting waste, this is our way of maintaining water balance. This is our way of regulating blood pressure. The urinary system works really well specifically for that. We'll be able to look at that in this last and final unit. So what I plan to cover today, I plan to cover just the basics to take a look at, again, giving you kind of an anatomical perspective of the kidneys. We'll look into the cross-section of the kidneys and show you where specifically the functional unit takes place. We'll dive into the functional unit, understand the mechanisms behind the nephron, which is our functional unit, point out its role, and then as we go through the nephron, we'll start to understand how we can create urine, we can reabsorb stuff from the urine, and then secrete out any additional wastes. And I'll break each of those down for you. Aim today is to really just cover the anatomical portion and talk about the first function in the nephron. So altogether, what is the role of the urinary system? And the urinary system is basically all for maintaining homeostasis. Not only are we going to be looking at water levels that will regulate our blood volume and blood pressure, but we'll also be able to regulate our pH through the elimination of acids, namely our hydrogen ions. And then also being able to regulate the tonicity of our blood, mainly through adjustment of the osmolarity of the blood. And if you remember, the osmolarity is tied into the amount of solutes in a solution. So we're looking specifically at the stuff that's dissolved in the blood. And what stuff do we have in terms of electrolytes would be sodium, we have our potassium, we've got calcium chloride, we've got phosphate in there, magnesium, and bicarbonate. All these things we need to be able to maintain normal homeostatic values. And again, ever looking at blood labs, you saw the reference values for each of the components in our blood labs. There are also urinalysis that's also done as part of a panel to determine the level of the ions in our urinary system. We'll take a look at how the kidney also reabsorbs glucose. And why are we reabsorbing glucose? We're pretty much dumping fluid in our functional unit, our nephron, and then we're going to have to reabsorb some of that back. And then at the same time, eliminating excess wastes that are also present in our blood. And then we'll also look from a hormonal perspective, the other hormones like renin and erythropoietin, EPO. We don't really talk about the last one, EPO, that much, but nonetheless, we'll be looking at the renin, angiotensin, aldosterone system. So looking at the kidney, here you can see the kidney. We've taken a frontal slice, and what's prevalent in this particular slice is that you can see two distinct layers. You'll see the renal cortex, which is the outer layer, and then you'll see the renal medulla, which is the inner layer. Now, what you're going to be able to see, if you pay a little attention over to the cortical layer, is you're going to see this functional unit, the nephron, and you can see where that's nestled in. So on the cortical layer, you'll be able to see the formation of our dilute urine. And we'll go into specifically the function of that. We're going to be looking at how we create this dilute urine through the blood vessels surrounding as well as the nephron. You'll understand the system behind that. We'll cover that today. And then when we look at the medullary function, when we move down into the medulla, you'll see how we're going to be able to take that dilute urine and make it much more concentrated. And we do that because we're reabsorbing some of the stuff as it makes its way down. So where you're going to see the formation of the urine, you're going to see it's going to travel down this yellow tube, this collecting tubule, and making its way down to this renal pelvis here, which is the center. And this renal pelvis is the intersection where you're going to see blood flowing in, blood flowing back out, and the collection of this urine that will make its way over to the bladder, down to the urethra, and out into the external environment. So within the renal medulla, you'll be able to see different functional areas. And you'll see different pyramids. These are these pyramids that are in the shape of cones. And these are going to contain the apparatus that are involved in the formation of this of urine. And then in between these pyramids, you'll see these columns. And these columns are going to contain this fibrous material that's going to anchor the cortex with that of the renal pelvis. And then you're also going to see the flowing of blood, both arteries and veins, through these sections. So really, the functional area that's going to be involved are these pyramids. You have these different, and it varies from person to person, but about 8 to about 18 of these subdivisions that are present. Now, as that as the urine comes in and you get this the concentrated urine, this is going to flow down into these ureters, and these ureters are going to collect into the bladder. And the bladder is going to be able to store that concentrated urine. And so what we're going to be able to see within the bladder is that the bladder will start to fill up. As the kidneys start coming in, dumping in this concentrated urine, you'll see that this urine will start to fill up. And we do have transitional epithelium, which we discussed in our tissue lecture. And transitional epithelium consists of both simple squamous, or not simple, I'm sorry. It'll be stratified squamous layers where some will be squamous, some will be cuboidal. And you get multiple layers where it accommodates the stretchiness of that particular organ. So as these fill up, there's going to be, the next section which we'll be able to see, is as this starts to fill up, this this bladder can hold quite a bit of fluid. So upwards about the 700 milliliters to about a liter of urine. And how we eliminate this urine is through a process called urination, the technical term being micturition. So we notice that there are stretch receptors that are present in the bladder, in the muscle layer of the bladder itself, and specifically more so in the sphincter located down here. So as the pressure starts to increase with the bladder filling up, there are stretch receptors that send signals over to the pons, specifically to this area. It's the spinal area called the pons, where there's a micturition center that receives input involving how much stretch there's taking place in the bladder. And as that fills up and that applies, as that pressure comes in, or the stretch receptors pick up information that's sending over to the pons, the micturition center then sends information back, letting us know to release that urine. So this entire reflex right here, where we get the initial stretch, comes back, sends information back, and causes a release of urine. And this is actually exhibited under voluntary control. We'll talk about this towards the end of this lecture, where the voluntary release of micturition or urination is how we empty the bladder. That emptying of the bladder will result in less stretching that takes place, causing a negative feedback loop. Looking at the urethra, everything from the bladder out to the urethra is how we eliminate our urine. In males, it's also the way that we eliminate semen as well. Now, let's zoom back into the kidney. Again, we'll reserve the process of the formation of urine towards the very end of this lecture. I want to go back and focus back specifically on the kidneys itself. And first thing we need to look at in our kidneys is to look at the blood flow and how we get blood into the kidney and then how we pull blood back into out of the kidney through the renal vein. So renal artery is where we start. The renal vein is where blood comes right back out. And then looking at kidney transplants, really the main three areas that you would see for a kidney transplant, really the only connections that need to be made are going to be your renal artery, your renal vein, and then the ureter. And so technically, you can see that we can still live normal lives with one kidney. So if you're ever to donate a kidney to someone who needs it because you need a match, you can still live fairly normal lives. In fact, I've been told, and I haven't been able to verify this through any journals, that really we only need 1/3 of a kidney for at least normal functioning. Okay? But this is going to play a major role when we start to increase cardiac output. That can affect the amount of the level of kidney functioning. So as long as assuming that everything is fairly normal, we have a lot of redundancy in how much we need. And in fact, if you were to ever donate a kidney, or if you ever lose a kidney for any reason, what would happen to the other kidney? It would enlarge, and it would assume additional function. So the purpose of the blood flowing through obviously is going to be for the process of filtration. How much of it is going to flow through? You'll be able to see that most of the cardiac output, we're going to get a lot of filtration that will take place in the kidneys. We'll see how it takes place in the renal artery, which is going to be a branch off the descending aorta. And down the descending aorta, the renal artery will come in and supply blood to the kidney for the process of filtration, and then blood will return back through the renal vein. So in that process, let's start from the renal artery and end up at the renal vein. And we know that blood flows through these renal columns. So let's see how we get from the renal artery to the renal columns into the functional unit and back to the vein. And that'll help you understand the blood flow. The second component we need to look at later on is what happens to the fluid that's pushed out of the blood into our functional unit, and how we claim that a good chunk of that fluid back. So we start from the renal arteries. The renal artery is going to come in and branch apart, which we're going to see the segmental artery. And from the segmental artery, we're going to move in between the lobes, in between each of these little conical pyramids. And these are going to be called the interlobar arteries. Okay? So in between the lobes. The interlobar arteries will then branch out. They'll curve around, and they'll become the arcuate arteries. So if they curve around the base of the pyramid, it'll be called the arcuate artery. From the arcuate artery, we're going to branch off into what's called the interlobular artery. Not the interlobar artery, but the interlobular artery. And those are going to be branches off the arcuate artery. From the interlobular arteries, we're going to see now, namely, our functional unit. In this area, from the afferent arterioles over to the peritubular arteries, are going to be the blood supply for the nephron. And we're going to move from the afferent arteriole. We're now going from our arteries to our arterioles. The afferent arteriole will turn into our glomerular capillaries. And from our glomerular capillaries, we're going to branch off again into the efferent arterioles. So we're going from arterial back to capillary to arterial. The efferent arterioles will then move down. You'll see the peritubular capillaries, and I'll show you these when we zoom in. Peritubular capillaries will converge back into the interlobular veins, and we're going back the reverse down the renal column. We're going to go from interlobular to arcuate to interlobar veins, back to the renal vein, in which we're going to get filtered blood back into our systemic circuit. So looking at, and again, we'll review these arterioles, capillaries, and arterioles. We'll zoom on this when we look at our nephron. Let's take a look at the other functions of the kidney, or our main functions of the kidney. As I mentioned earlier, the excretion of metabolic wastes. The waste that we'll be seeing are things after the metabolism of specific monomers. And so looking at the things that we were able to break down, we were able to break down our main organic molecules. We've turned our proteins, we've broken down our proteins down to amino acids. We've taken our DNA and broken them down to our specific nucleotides. We had our starches, broke them down to glucose. And I'm missing fats. We've broken our fats into fatty acids. So here we're going to be able to see metabolic waste. These are things that are involved in cellular respiration. Or actually, sorry, I take that back, not cellular respiration, but in metabolism. When we break down amino acids, one of the byproducts of breaking down amino acids is urea. And urea is produced by ammonia, which is one of our main byproducts from breaking down amino acids. So if you remember our amino acids, we had NH2, C, and then this was a carboxylic acid. And then from here, we had our molecule. And then on top, we had our R group for that particular amino acid. And in the consumption of amino acids, when we break it down, we're going to be able to release this amine group, and that amine group can then join hydrogen ion and create NH3+. And that is ammonia. And ammonia combined with carbon dioxide creates a byproduct called urea. And you'll be able to see how we eliminate urea in the kidney. Creatinine is the breakdown of creatine phosphate. And we haven't talked about creatine phosphate because we didn't really get into muscle physiology. But one of the ways that we were able to store in the muscle, we're able to store ATP, is through creatine phosphate. So this is our storage for creatine phosphate. And whenever we break that down, we break apart creatine phosphate, we create something called creatinine. So for any of those that do use any supplements, does anybody use creatine monohydrate here? Yeah. Creatine is exactly what that product is for. It's to increase your storage for ATP in the muscle. Out of all the supplements out there, that was probably the most effective. And if you look at all journal articles that examine most of the supplements out there, caffeine and creatine are typically the most useful, best bang for your buck. And I'm not really trying to... What's that?
And they're the cheapest.
They are the cheapest. And I'm not trying to advertise it. I'm not saying that you should use them. I'm just saying, in general, one of the things that's a big thing about people that take in creatine phosphate, and really just applies for everybody as well, even though we have normal levels of creatine phosphate, the breakdown of creatine, we break it down into a product called creatinine. And creatinine is something that the kidneys need to get rid of. So typically, creatinine is something that we can measure in the urine to determine kidney function. How fast do we break down, given the amount of normal levels of creatine phosphate, how fast can we get rid of creatinine? And there's certain oncology protocols, certain treatment protocols that will look at kidney function, and they base off that how much creatinine is present in the blood. Okay? Present in the blood and present in the urine. The last of these is going to be uric acid. And uric acid is something that is broken down with the breakdown of nucleotides. So we break our DNA down to nucleotides, and once we break down those nucleotides, we have uric acid. And for the most part, it is insoluble in the blood. It is concentrated, and it concentrates mostly in the joints. Does anybody here have gout or know of anybody with gout? Gout is basically an accumulation of uric acids. And our inability to eliminate the accumulation of uric acid results in a condition called gout. And so people that have gout, if you ever hear of anybody, especially in their 30s, maybe late 20s and 30s, which I know a bulk of you are probably entering that age, if you ever start having pain in your right toe, like right at the main toe, more often than not, it's probably either it's an acute injury if you know you injured yourself, but if you didn't injure yourself and you start to get that pain, that's the start of gout. And especially you'll know it after a bout of drinking and red meat. And basically what they'll tell you is to eliminate these foods because you're starting to show symptoms of gout. I know it because I was told that I need to start avoiding it. But hell no, I'm not avoiding it. He gave me a whole list. I remember the doctor telling me, so gout's not that bad. You just got to eliminate some foods out of your diet. And he starts looking through his list. He's like, yeah, you got to get rid of You have to get rid of beer, you have to get rid of red meat, you have to get rid of cheese. And I'm looking, I'm like, are you serious? And he's just like, yeah, no, that sucks. I'm sorry.
Just end it there.
What's that?
Just end me.
Yeah, no.
Just go home with some allopurinol and some coaches.
I'm fine, I'm good enough. And if I ever have any, there's prophylactics you can take, and you're not taking away my red meat and my beer. That's going to be a decreased quality of life for me. The other portion of the kidneys, this is our way of eliminating wastes through our urine. The other portion is to also look at our water and salt balance. And all this is going to be related to normalizing blood pressure and blood volumes. Are we going to be able to get rid of excess water if we have it? We're going to be able to get rid of excess salts if it's present. So you'll be able to see this process as we look at the nephron, and you'll be able to see the nephron. You'll see these tubules, and then this descending tubule down over here, which we'll point out later. This whole process is the formation of a filtrate. And again, we're going to go over this today, and how we're going to be able to exchange with the tubules that are wrapping around. The tubules are going to be the capillaries that are going to come in and collect the stuff from the urine that was formed. It's going to collect it and either collect it or not collect it. And this is our way of getting rid of excess stuff. The stuff that's coming out, this portion back right here, is what we're doing when we're making our way back towards the ureters. This is our way of eliminating waste. So this reabsorption process, if we've got too much of something, we'll be able to eliminate it. If we've got too little of it, we'll be able to conserve it. So if you ever have a very salty diet, let's say you have just eaten a really salty broth and you've taken a lot of it out, you notice that after an hour or two, whatever you're eliminating, you may notice how salty your urine is. I don't know how you can really figure that out, but you can at least, I don't know, maybe smell it. I don't know what you would do, but you can tell the composition's a little bit different. This is our way of eliminating excess. You know that if your blood pressure is low, if your blood pressure is low, we have mechanisms in place to be able to conserve that water. And ways we can conserve water is to increase thirst. You can also eliminate sweating. And then the other portion is to use antidiuretic hormone. Antidiuretic hormone will help you conserve water. So essentially what we're going to be doing is we're going to be reclaiming salts, and then we'll be using antidiuretic hormone to allow us to reabsorb water. So we're going to be involving the reabsorption of water and salts, or the elimination of those two, in order to maintain homeostatic levels of our salts as well as our water volume. Acid-base balance, this is our way of maintaining normal balances of hydrogen ions, and namely our bicarbonate ions. So by now you know your bicarbonate ions are used as a buffer to keep our pH between 7.35 to 7.45. I know all of you guys know that because 100% of you guys got this right on the test. So the whole purpose of this is our way of reabsorbing bicarbonate ions to assist in the buffering capacity of our blood. Anytime we end up with excess hydrogen ions, either through metabolic acidosis or anything like that, we're able to eliminate excess acids through the urine. And there's the mechanisms which we won't really get too much involved, but our way of our ammonia that's been produced through the breakdown of urea. We combine that with hydrogen ions. We can eliminate those, but at the same time eliminate excess hydrogen ions through that process. And then the secretion of hormones. We still have to look at the hormonal functions, where we're going to see renin as one of the enzymes in the kidneys, which is going to be playing the initial role in the RAS system, our renin, angiotensin, aldosterone system. Okay. And through this process, we would see our ability to maintain salts as well as volume of H2O. Looking at our water balance, I do want to point out, again, looking at the cell. And in the cell, we're going to have our intracellular fluid, and then the water on the outside is going to be our extracellular fluid. And as a recap, we know that the bulk of our extracellular fluid is going to consist of blood flowing through the capillaries, so these are going to make our capillaries, as well as the interstitial fluid surrounding. And within the intracellular fluid, we know the ions that are present are going to be potassium. On the outside, we're going to have glucose. We'll have salt, our sodium. We'll have also chloride. We'll also have calcium. We also have phosphate and magnesium involved in the inside, which is not really something that we talked about too much. We've got tons of bicarbonate on the outside. So given all of these, we need to be able to maintain normal values of potassium, phosphate, and magnesium within the cell. And so in order to maintain those, we're going to have to exchange with the external environment. And that exchange of the external environment is essentially going to make its way back into the kidneys to eliminate any excess of. So I know one of the quiz questions that I asked you prior was what happens with excess potassium ions. Excess potassium ions that we're not able to eliminate through the urinary system can have a profound effect in terms of the membrane potential of the cell and can affect the firing of the cell itself. So in those cases, we want to make sure that salt levels are normal within both the intracellular as well as the extracellular fluid. The other portion that we'll be looking at is, again, another recap, is to look at the movement of fluid. So we're going to once again take the capillary bed, which I'll draw as a straight line. We have our arterial side, and we have our venous side of the capillary. And we knew through this process, if this were the capillary, we knew that we had a couple pressures in play. We had our blood hydrostatic pressure. Blood hydrostatic pressure is the pressure of the blood within the capillary exerting pressure out. So it wants to push against the endothelium and push out. So this is hydrostatic pressure. Now the other pressure that was involved was blood colloid osmotic pressure. And blood colloid osmotic pressure was the pressure of blood moving, the tendency of water wanting to move from low concentration over to a high concentration. So technically what's happening with blood colloid osmotic pressure is the presence of proteins and red blood cells that wants to draw water from a hypotonic solution over to a hypertonic solution. So to draw this out in terms of our U-tube example, okay, here's our semi-permeable membrane right in the middle. We have our solutes that are present on the left side, and we had our two solutes available on the right side. Osmosis is the driving of water from our hypotonic solution over to our hypertonic solution. So we know that the hydrostatic pressure is over to the right. Osmotic pressure, this would be our BCOP, this would be our BHP. The osmotic pressure is the pressure of water movement from the hypotonic side over to the hypertonic side. Blood hydrostatic pressure is due to the buildup of all that fluid on the left side. Now you have this hydrostatic column. This is going to exert a pressure of pushing water back the opposite way. Now, accounting for all these pressures, we know that the movement of water, bulk flow, that takes place on the arterial side, due to the higher pressures of blood hydrostatic pressure, compare that to that of blood colloid osmotic pressure, we're going to get more net pressure, net filtration pressure, taking place on the arterial side, hence filtration. And due to higher colloid osmotic pressure compared to that of hydrostatic pressure, we're going to get reabsorption. So these pressures that are involved that will result in filtration and reabsorption. The direction of fluid movement we're going to be able to see in the kidneys. So given that, so now we've talked about the movement of water, and then as well as all the stuff that might be dissolved in the water, let's consider those aspects for water input as well as water output. Water intake is driven by the thirst center. So thirst center in the hypothalamus, we do have osmoreceptors that are present that will give us the pressures within that fluid. And the osmotic pressures that are exerted within the fluid that flows through will tell us, and that osmotic pressure again is due to the solute concentration, will drive our thirst. So the saltier the water content in our blood, the increase in the osmoreceptors firing back over to the hypothalamus. The hypothalamus will then, specifically the thirst center, will drive then the need to drink water. So this is going to increase our water intake. So as we start to increase our water intake, that's going to cause distension of the stomach. And the stretch receptors in the stomach will then feed back into the thirst center, letting us know that we've drank enough water. So think about that in terms of how this can be confused for hunger. Because you probably have heard at some point, if you feel hungry again, it could potentially be because you're thirsty. And for those of you that might eat something and still continually feel hungry, well, what's technically happening is that thirst is driven due to the high solute concentration in the blood, which will drive the thirst center. And the thirst center will let you know that you do need to take in water. But take in food, and that will also cause stomach distension. That stomach distension will then inhibit the thirst center, but at least temporarily. But that has not resolved the osmoreceptors still firing back, letting you know you're thirsty. You're temporarily inhibiting that process through stomach distension. So try that out at some point. If you're ever feeling hungry, or if you're, I don't want to let you know just to drink water if you're feeling hungry, but for the most part, if you're ever, if you notice that even though you've eaten, you still have that feeling of hunger, it might really just be your thirst intake. The output, so we're taking in water through the alimentary canal. The output, mechanisms for water loss, 60% of that is going to take place through the urine. Defecation, a very small amount of water through defecation. And then the bigger chunk, or second to biggest chunk, would be through the skin and lungs. We eliminate water through the skin through sweating, and lung through water vapor that's lost through exhalation. And our way of being able to regulate all those, our output, is specifically looking at the use of antidiuretic hormone. We'll look at ADH, and to a certain extent we'll look also at angiotensin II as well as aldosterone in maintaining normal osmotic pressures. Again, these are going to be related to the salts as well as the volume of H2O. So with that, we've now talked about specifically the functions. We talked about the blood flow. Now we need to take a look at the nephron. So with the nephron, you'll be able to see this is the functional unit. And again, you're going to be able to see the divisions of the kidney. We have our cortex as well as the medulla. So in the cortical layer, you're going to be able to see different parts of the nephron. And we start off with the glomerulus. You're going to see these tubules, and those extend out, travel down into the medulla, come right back up to the cortex, and travel again right back down into the medulla for the creation of concentrated urine. We do have a set number of nephrons in the kidneys, and that doesn't change. The blood vessels that we'll be able to look at, we'll be able to look at the afferent arteriole, we'll look at the capillaries, and then we'll look at the efferent arteriole. and then all the capillaries that come in and wrap around the tubules. If we were to straighten all this out, you'll pretty much, just to simplify, you'll see the nephron, where you're going to see two different areas of the nephron. The renal corpuscle, which is going to be the head of the nephron, which you can see in in this process right here. This would be our renal corpuscle. And if I were to straighten out this tube and make it one straight tube, these would be considered our renal tubule. And this is everything down from this point on. So the renal corpuscle will be able to look at the main components of the corpuscle. We have our glomerular capillaries, which are going to contain the blood. And the glomerular capsule, which will contain our filtrate, which is going to be our dilute urine. It's what we first start off with. So we have to understand the interface between these two for the process of filtration, where we're going to have fluid escape, the glomerular capillaries, and all the stuff that was filtered out of the capillaries are present as filtrate. That filtrate is going to move down the tubule, and the tubule representing the longest portion. This tubule, what we're going to be able to see is that we're going to see what's called reabsorption. Reabsorption is what we're taking out of that dilute urine. There's a bunch of stuff in that urine that we need to recollect, namely water, some ions, as well as the glucose that's present. We'll talk about what's actually present in that filtrate on the next couple slides. We're reabsorbing stuff that we need, and then anything that we need to get rid of, we're going to be, this is going to be called secretion. This is stuff that's going back from the blood back into the urine. And reabsorption as well as secretion all take place in different areas, and you'll see where those areas specifically take place. So let's zoom in specifically into the renal corpuscle, again, looking at the glomerulus as well as the capillaries present within. The renal corpuscle is composed of our capillaries. The capillaries, again, are between the afferent arteriole. Here's our capillaries that are all bunched up together, and then the efferent arteriole. So we're looking specifically at the capillaries, and then the capsule that's wrapped around. So in that capsule, you're going to see what's also referred to as the Bowman's capsule. The glomerular capsule is going to be a double-walled epithelial capsule that comes in and surrounds these capillaries. And I'll talk about where those double walls exist. You'll see the formation of filtrate as it comes out and forms out and collects in the glomerular capsule and then makes its way down the rest of the tubule. And the first stop will be the proximal convoluted tubule where reabsorption takes place. We're going to have the efferent arteriole. Here's our efferent arteriole. That's going to come back in, and that's going to start wrapping around the rest of those tubules, coming in, collecting all the stuff that's present in our initial filtrate. Looking at the glomerulus, here you can see another picture of it. You can see the afferent arteriole. This is blood entering. You'll see the capillaries that are present within, and you'll see there's this whole formation of stuff wrapped around, and I'll talk about that in just a bit. And then you'll see the efferent arteriole. One thing I want to bring up, you're also going to see something I want to point out later, is the distal convoluted tubule. And let me go back to this earlier slide right here, and let me just erase this bit right here. One thing that you'll notice about the tubule, here's our glomerulus. We have the proximal convoluted tubule, and it's called the proximal convoluted tubule because it's convoluted, it's curvy, and it's the closest to the glomerulus. After we leave the proximal convoluted tubule, we'll have what's called the loop of Henle, which dips down. This is going to be the straight trombone-looking like portion of the unit that will dip into the medulla. It'll come right back up. So this whole thing is called the loop of Henle. It loops back into the medulla. We have the descending portion of the loop of Henle, and we have the ascending portion of the loop of Henle. Once we come back, you're going to see the distal convoluted tubule. Again, convoluted because it's twisty, and distal because it's the furthest away from the glomerulus. That distal convoluted tubule comes right back up right next to that glomerulus. which you can see back in this later slide. Distal convoluted tubule comes back up. This area, there's a specific area that we'll reference later called the macula densa, which are going to contain chemoreceptors that will give us feedback about the stuff that's present down the line from the proximal convoluted tubule, down the loop of Henle, back up to the distal convoluted tubule. This will provide good feedback into the glomerulus to let us know how well this whole nephron is working. So pay attention to that when we get to that section. Distal convoluted tubule comes right back up, and then we've got the collecting duct, which will help extract more water to make it down back as concentrated urine back into the renal pelvis. So, coming back to that, you'll see the distal convoluted tubule coming back up. The other portion that I also want to talk about are what are called mesangial cells. And that's really it. So this area, the capillary, is going to be where filtration occurs. As I mentioned earlier, filtrate is going to be pushed out of the capillaries and will flow down the rest of the tubule. So what's going to be present in our filtrate, again, we'll talk about in a couple slides. So this process I talked about a little bit earlier in an earlier slide. This is, again, just talking about the afferent arteriole to the capillaries, back out to the efferent arteriole. These, from this point on, past the efferent arteriole, we're going to have what are called peritubular capillaries. When you get to the book, you're going to see a section of the peritubular capillaries called the vasa recta. Let's see if I have that slide somewhere. The efferent arteriole. Here you're going to see these are going to call the peritubular capillaries. And the capillaries that wrap around the loop of Henle are commonly referred to as the vasa recta. So if you ever see those terms, it might be a little confusing at first. It's just the vessels surrounding the peritubular capillaries. And again, the importance of the vasa recta as well as the peritubular capillaries is to reabsorb the stuff in the filtrate that we need to keep. So going back, let's now focus specifically on the renal corpuscle. And looking at the capillaries, as we mentioned earlier, let's also look at the capsule surrounding, the Bowman's capsule. So in this glomerular capsule, we do have a double layer. And that first layer is going to be the parietal layer, which is the outer layer. This outer layer is present that wraps around the glomerular capillary, that wraps the outside of the glomerular capillaries. The visceral layer, you can't necessarily see it on here, but the visceral layer is flush right against the glomerular capillaries. And the whole purpose of this visceral layer is to provide a covering for those capillaries. So if you take a look at the visceral layer, notice these dots right here? These dots are called podocytes. They're cells that are part of the internal layer, that visceral layer. And these podocytes have these extensions of the cell membrane. And they literally will wrap around. And you might not be able to see it on this slide because of the color scheme, but you'll definitely see it on this slide over. Here you can see our podocytes, and you can see the extensions of the cells. And you notice how these cells create these slit-like layers? These slits right here are the grates that come in and serve as a protection on the outer layer of the capillaries. So what I want to bring up here, so as I mentioned, the filtrate is going to be forming out of, you've got fluid moving out of the capillaries. What type of filtration takes place? Because again, this is where filtration takes place. What type of filtration takes place? We have multiple layers that assist in keeping bigger objects within the capillaries. Things that need to stay in the blood are formed elements. You want to keep your red blood cells in there. Things that are dissolved in the plasma. What's dissolved in the plasma? You've got some large proteins. You've got water-soluble hormones. You've got glucose. You've got ions. And you've got tons of water. So looking at the structure of the capillaries as well as the podocytes on the outside, those podocytes, as I mentioned earlier, have these extensions, these finger-like extensions called pedicels. Pedicels are these fingers that come in that literally will lock together like this. And the slits in between my fingers are exactly these filtration slits that you see. So the capillaries are technically fenestrated capillaries. They contain a bunch of pores that allow things that are dissolved in the blood plasma to flow through. Namely, there are proteins, large and small, all the glucose, ions, and water. You also have this basal lamina that's just going to be on the outside of the endothelium that will prevent larger proteins from flowing through. You're going to have these slits that are going to prevent medium proteins from flowing through. And again, the grates right here, the fenestration, the pores, will maintain all the red blood cells and all the other cells contained within the blood inside. So this really helps filter out only plasma with small proteins, really small proteins, but namely things that are dissolved within, our glucose, our ions, and water. And again, this is stuff that is filtered out. So you get the bulk of water, glucose, and ions making its way out. This stuff, most of this we want to reclaim back, and we're going to be able to reabsorb that in the process called reabsorption. So these are the contents and stuff in filtration. Some of the other things that we can note in an abnormal function of the kidneys, if you note that any dysfunction of these pedicels, or let's say any mutation of the proteins involved in creation of these pedicels, or any malformation of the fenestrations on here, or the basal lamina, can cause openings that are bigger, and that'll allow larger proteins, that could allow the leakage of red blood cells, and that causes dysfunction of the kidney itself. Now as that filtrate moves out, it's going to enter the space within, between the visceral and the parietal layer, and that filtration that comes in, as I mentioned earlier, will flow back down the tubule. So we're going to have to take a look at the pressures involved. We're not going to look at it just yet, but the pressures that are involved are the two things that we looked at. Blood hydrostatic pressure, colloid osmotic pressure. The other new one that we need to look at is capsular hydrostatic pressure, and I'll talk about that in just a bit. So the process for filtration to take place, like you learned earlier, are the pressures that allow filtrate to be produced. This is going to be net fluid pressure out of the capillaries that will push fluid down into the proximal convoluted tubule. So as that fluid flows through, I mentioned the proximal convoluted tubule, the loop of Henle, the distal convoluted tubule. Not all nephrons have the same length of the loop of Henle. There are some that extend further down into the medulla, and there are some that don't even really reach the medulla. So various lengths of the loops of Henle. And typically, general rule is, the longer the loop of Henle, or the longer the pathway of the tubule, the more concentrated the urine becomes. Some additional structures that I do want to talk about that are also going to play a major role are juxtaglomerular cells. And the juxtaglomerular cells are present in the afferent arteriole. That's blood flowing into our glomerular capillaries. Juxtaglomerular cells, or otherwise known as JG cells, are part of this apparatus that detects the blood pressure, particularly within the afferent arteriole. Because what we need to do is we need to maintain normal pressures within the glomerular capsule. Because if we can go back to an early example of what I looked at when we pinpointed the capillaries, I pointed out what would happen if hydrostatic pressure was low. And in the cases where hydrostatic pressure was low, if you remember In our regular capillaries, we had a blood hydrostatic pressure of 35, 25, and then 16. And then our blood colloid osmotic pressure had a pressure of about 25 millimeters of mercury. That was normal pressures. What would happen if blood hydrostatic pressure decreased? Let's bring it down to 25 millimeters of mercury, 15 millimeters of mercury, and then 6 millimeters of mercury. What we ended up with, no filtration. Most of what we saw was reabsorption. So no filtration. And now take that back to our kidneys. If I don't get normalized blood pressure within the glomerular capillaries, the process of filtration will not take place. And if I'm not filtering in the glomerular capillaries, the kidney isn't doing its function with what it's supposed to be doing. It's to create dilute urine and reabsorbing what we need. So that would be, in essence, a form of kidney failure. So in this process, we're going to be able to maintain, and with these additional structures, this is our way of maintaining normal blood pressure and allowing normal filtration to take place. So the JG cells are present within. There's these cells contained within the JG cells that detect pressure. So there's stretch receptors that are located within the smooth muscle surrounding the afferent arteriole. And I'll talk about that at the end of today's lecture. Mesangial cells are present in between the capillaries of the glomerular capillaries and right before the capillaries. There's two different types of mesangial cells, but for the most part, they work the same way. Mesangial cells, think of them as a net. You've got the afferent arteriole, and then you've got all these capillaries that are present within the glomerular cup or within the renal corpus. In order to decrease the surface area to slow down the filtration process, I can retract some of those capillaries. If I want to increase filtration, I can open up those capillaries up even further. So these mesangial cells help contract and relax to either pull back the capillaries or allow them to fully expand. The other portion is called the macula densa. And the macula densa are chemoreceptor-filled cells that are part of the distal convoluted tubule. And they come on and they provide feedback that, to the juxtaglomerular apparatus that will help regulate flow of blood into this area. And what the macula densa specifically detects is the salt concentrations. They're looking specifically at sodium as well as chloride ions. So this is our indirect, which I'll talk about later. This is our indirect measure. This is our direct measure of blood pressure. This is our indirect of blood pressure and blood flow. So tying this all together, we have, once again, our nephron, which is going to consist of our renal corpuscle as well as our renal tubule. And then also the blood supply that we mentioned earlier in the very first beginning. Everything from our renal artery feeding into the afferent arteriole, glomerular capillaries, efferent arteriole, and the peritubular capillaries surrounding the rest of the tubule. That comes in and collects right back into our veins to feed back into the returning systemic circuit. What questions do we have about blood flow versus the formation of the urine? So we're going to just talk about, after this portion, we're going to talk about specifically the glomerulus to start. Any questions? All right. So glomerular filtration. I talked a little bit about earlier, but as I mentioned earlier, the afferent arteriole is going to be feeding blood into the glomerular capillaries. And in that glomerular, as that blood is flowing from the afferent arteriole to the glomerular capillaries, you're going to see the pushing out some of the filtrate out. And that filtrate is going to be stuff that is present in the capillaries that'll make its way into the glomerular capsule, but not all the plasma from the blood is going to be filtered, meaning that we're going to see blood flowing through, we're going to be able to push some fluid out, but the rest is going to make its way down the afferent arteriole. So this is our first portion, again, called filtration. And that filtration is the primary process at which the kidneys function to filter the blood out. And how much of the how much filtration actually takes place? It's about 150 liters per day in females. It's about 180 liters per day in males. So if you take into account about 5 liters of blood, that comes to about 10 times amount of filtration of that blood. We're filtering your blood at least 10 times per day. So when you think about this filtration that's provided by the kidneys, and take into consideration people that have kidney failure, they typically would have to undergo what's called dialysis. A dialysis machine is something that they need to connect to in order to filter their blood manually. So typically in those cases, they'll be able to tap into an artery and tap into a vein. Blood will flow out the artery, flow into a machine that will manually filter out the blood to feed it back into the vein and remove those components manually. So with that, when we think about how much fluid flows out in filtrate, again, about 180 liters flows out per day. As that fluid makes its way through, we're going to be able to reabsorb that fluid. Imagine peeing out 180 liters per day. That's a lot of fluid. We want to be able to reabsorb about 99% of that. And as we reclaim about 99%, so 99% reabsorption, how much urine output are we going to be able to see a day? About 1 to 2 liters of urine, concentrated urine. So all the stuff that we don't want with that leftover fluid of water is urine. So looking at these, I'm going to talk about the pressures on this slide, but all the information that you need is available on the next slide over. So the 2 pressures that we talked about already, blood hydrostatic pressure and blood colloid osmotic pressure. The pressures are going to be slightly different in here because we're looking at the pressures in the arteriole into the afferent arteriole into the glomerular capillaries. And the blood hydrostatic pressure, the pressure in that area, is going to be 55 millimeters of mercury. Again, blood hydrostatic pressure within the arterioles into the capillaries. Blood colloid osmotic pressure, again, is the osmotic pressure. That's due to the presence of all the proteins and red blood cells. Water wants to move from the hypotonic solution on the outside of the capillaries into the cell. And that blood colloid osmotic pressure is 30 millimeters of mercury. It was 25 that we learned about in the systemic capillaries. It's about 30 in the kidneys, or specifically within the glomerular cup. So 55 and then 30. The other thing that we need to talk about is the capsular hydrostatic pressure. And what exactly is a capsular hydrostatic pressure? You have fluid exiting the capillaries. And it's not just freely moving around, it's actually collected in the visceral layer. Okay? That visceral layer of that visceral layer on the outside is going to start to fill up, and it's going to increase the pressure in the capsule. So that pressure, we've got pressure on the inside due to the blood, we've got pressure on the outside due to the formation of filtrate. That pressure on the outside is going to counter that of the glomerular hydrostatic pressure. So if I've got pressure pushing out to push blood out, I've got another pressure forming within the Bowman space that's wanting to push fluid back in. So the glomerular blood hydrostatic pressure, which is 55 millimeters of mercury, is going to be opposed by two pressures. Again, number one would be the capsular hydrostatic pressure, which is about 15 millimeters of mercury, minus 30 millimeters of mercury for the blood colloid osmotic pressure. So 55 minus 15 is 40. 40 minus 30 is 10 millimeters of mercury. And that is the net filtration pressure. That is the pressure, net pressure being exerted out to push filtrate or fluid out of the capillaries into the Bowman space to flow down into the proximal convoluted tubule. What questions do we have on the pressures?
You said for number 3 was hypotonic to...
Yeah, so think about what's present in the filtrate. It's mostly water, we've got glucose, we've got ions in there. In the blood, we've got red blood cells, we've got large proteins and small proteins. Because I've got more stuff dissolved in the blood, water is going to want to move from a hypotonic, this is hypotonic compared to this, which is hypertonic. So water wants to move in that direction. And again, drawing out that YouTube example earlier, I've got more solutes on my left side, and I've got less solutes on the right. The direction of water movement is towards the left. So the left represents the capillary. On the outside represents the Bowman space. That's filled with filtrate. So this pressure right here represents blood colloid osmotic pressure. The pressure out to the right is our GBHB, that's our glomerular blood hydrostatic pressure. The other pressure that's also involved is capsular hydrostatic pressure. And that's due to the pressure pushing back to the left as well. And again, that's the pressure built up of fluid on the outer, wanting to push right back in. Does that help? What other questions do we have? All right, so that is our glomerular filtration rate. And each of these are described for you. Blood hydrostatic pressure, capsular hydrostatic pressure, exerted against the filtration membrane by fluid in the capsular space, and then blood colloid osmotic pressure. So those haven't changed. All of these forces, the balance of all these forces, are Starling's forces. The same forces that you learned in the heart, same things about preload, and the expansion of the heart, or the filling of the heart due to preload, is going to cause a bigger contraction. Same thing with these forces. These are all the forces that are involved in the process of filtration. So given that, again, thinking about that amount of pressure, 10 millimeters of mercury for the net filtration pressure. That excess pressure, net filtration pressure, NFP. All those pressures over the course of the day cause this much filtration. So normal filtration, we'll be able to say for glomerular filtration, you'll see how much of this is filtered. We'll be able to show you what the normal values are. Looking at all of this, as you now understand the pressures involved, we know that the pressures involved are going to be fairly regulated. We want to be able to maintain normal pressures. And if our blood hydrostatic pressure is low due to decreased blood pressure or hypotension, we want to be able to regulate normal blood flow through the kidney. Otherwise, if the kidney isn't operating properly, we have malfunction of the kidney, and therefore we're going to have kidney failure at that point. So we need to regulate the GFR, the glomerular filtration rate, in order to maintain normal flow through the glomerulus. So how do we do that? How do we regulate the glomerular filtration rate? we can either adjust, there's two mechanisms involved. I can either adjust the blood flow in here, into this apparatus, the blood flow through the afferent arteriole into glomerular capillaries, to the blood flowing out through the efferent arteriole. And there are nozzles that I can use, and they're technically not nozzles, but I can vasodilate and vasoconstrict to increase and decrease blood flow. The same mechanisms that you learned in our vessel lecture in increasing and decreasing blood flow to the tissues, we can do the same thing to the kidneys. The other mechanism that's present, altering the glomerular capillary surface area available for filtration. And what I mean by that is how much of the capillaries are exposed to allow filtration. If I want to reduce the amount of filtration, I can pull them back and reduce the surface area of all these capillaries. If I want to increase filtration, I can open it right back up. So those are the mechanisms. These are the efferent ways of adjusting the glomerular filtration rate. And we'll be able to look at three main mechanisms, or at least three types of mechanisms. Renal autoregulation is related to the kidneys, and how we can regulate the glomerular filtration rate. So this is autoregulation. Why? Because the kidneys are regulating it themselves. This is an intrinsic mechanism. Kidney does it all by itself. The other two are extrinsic, meaning that they're going to involve external factors. Neural regulation is going to involve the autonomic nervous system, where we're going to be able to see parasympathetic and sympathetic pathways that will adjust the blood flow into the glomerular capillaries, as well as the glomerular filtration rate. And the hormonal regulation is going to involve the other hormones, namely angiotensin II, as well as atrial natriuretic peptide. Angiotensin II was our potent vasoconstrictor. And then ANP was our potent vasodilator. So again, these two have antagonistic effects with one another. We'll be able to see all these on the next slide over. Renal autoregulation. So we're going to talk about the reflexes involved. This is all feeding information back into the kidneys and the efferent pathway outwards to regulate renal blood flow. And the two mechanisms here are in place. The first one is called the myogenic mechanism. And the other second one is tubuloglomerular feedback. The first one, in a gist, involves blood flow, right? So this one involves blood flow. Tubuloglomerular feedback gives us, and blood flow would be direct in this case. This gives us ion, or let's just say, let's say ion concentration that will be able to infer the amount of blood flow. This is an indirect path. This is basically saying how salty is the urine, and depending on the saltiness will give us an idea of the amount of blood flow through the kidneys, or through the afferent arteriole. So the myogenic mechanism is entirely a negative feedback mechanism. Now think about what happens when, let's say you have a hose, right? You've got a hose, you've got a valve that's flowing water through that hose. You don't want too much water flowing through it, because then your hose is flopping all over the place and can cause damage to the hose itself. You don't want too little of it, because now you've got too little of the blood flowing through. You want to have just about the right amount. So technically, the glomerulus wants an optimal level. It doesn't want too high, it doesn't want too low, but can accommodate a good amount of flow through the mechanism, through the glomerular apparatus. So as you take that hose and you turn it up quite a bit, that pressure is going to stretch out the walls of the hose. And we don't want that to happen. So technically, what's happening in the afferent arteriole is that as blood flow, let me go back to an earlier slide. As you get too much blood flowing through, I've got high blood pressure and therefore high blood flow through. That's going to stretch out the JG cells, or juxtaglomerular cells. They'll stretch out, and there's stretch receptors that will pick out the level of stretch. So in order to reduce the amount of blood flow, that stretching will trigger the contraction of the smooth muscle cells. It'll come in, due to the stretch, will cause the smooth muscle cells to now vasoconstrict. And that vasoconstriction will reduce the amount of blood flow into that area. So what started off with too much blood flowing involves vasoconstriction, which will reduce the amount of blood flow. Does that make sense? The second mechanism is the tubuloglomerular feedback. Again, going back to our earlier slide, the macula densa is going to be involved. And the macula densa here is from the distal convoluted tubule that wraps all the way back up. And let me go back to an earlier slide. Here you can see the distal convoluted tubule coming right back up, right near the glomerular cup. And this section right here has these macula densa. The macula densa have these chemoreceptors that detect the salt concentration. And if the salt concentration is off, it can come in and cause vasodilation. And the vasodilation is provided by nitric oxide. So if I inhibit the amount of nitric oxide, which is a potent vasodilator, the vasodilator, if I inhibit the vasodilator, that's going to cause vasoconstriction. Again, all related to the ion concentration. And the mechanism is fairly complex, so I don't want to get into too much detail about it. But all it's really saying is that if the ion concentration's off, we need to adjust via nitric oxide. So that covers renal autoregulation. Again, regulation that's all done by the kidney all by itself. At no point did I ever say autonomic nervous system involved, there's no other hormones coming into play, the kidney can do it all by itself. Neural regulation is all through the autonomic pathways. And looking at these, the sympathetic fibers or sympathetic pathways release norepinephrine. We already established that these that the postganglion fibers that come in and terminate, so here's our postganglion sympathetic neurons, that terminate on the afferent arteriole. They're going to be releasing norepinephrine. We have adrenergic receptors on these smooth muscle cells. And those adrenergic receptors receive norepinephrine and cause vasoconstriction. If you can go back to an earlier slide, specifically when we talked about, and I believe that was our endocrine lecture, we talked about cholinergic and adrenergic receptors. Adrenergic receptors are the ones that pick up epinephrine and norepinephrine. Some cells respond by vasodilation. Some cells respond by vasoconstriction. Okay. Or rather not vasoconstriction, because it relates to the vessels. When we looked at the adrenergic receptors in the smooth muscle cells surrounding the bronchioles of the lungs, that caused dilation, so relaxation of the smooth muscles. The adrenergic receptors here for the smooth cells, when they're activated, cause contraction. So the contraction that takes place on the smooth muscle cells surrounding the afferent arteriole causes vasoconstriction. Why is vasoconstriction important? Because vasoconstriction also increases the blood pressure in that area. When we increase blood pressure, that's going to be able to maintain, or actually technically increase the filtration rate. So we want to be able to maintain, and the best case example is to look at what happens when we have decreased blood volume or decreased blood pressure. And a good example of that would be hemorrhage, loss of blood, or loss of blood volume. Any drops in blood pressure, we need to increase blood pressure in order to maintain normal kidney function. The last of these is to look at hormonal regulation. And the two hormones that are involved, angiotensin II and ANP. Angiotensin II being, again, the potent vasoconstrictor. And what that does is to reduce the global glomerular filtration rate. So not only are we constricting the input for the glomerular capillaries, we're also going to constrict the output of the glomerular capillaries. Meaning all I need to do is squeeze both of these sides, and that's going to reduce the amount of blood flow through the glomerular capillaries. In effect, reducing the GFR. Atrial natriuretic peptide, remember again, when blood flows through the atria of the heart, is going to release ANP. So ANP comes out and causes relaxation. Where does ANP take effect? And I did talk about ANP as being a potent vasodilator. It specifically works on the mesangial cells. And again, the mesangial cells are present in between the capillaries. So that's going to increase the surface area. It's going to open up as much of the capillaries as possible to increase the filtration rate. So this raises the GFR. This reduces the GFR. Again, another mechanism as to how these are antagonistic hormones. One was a vasoconstrictor, the one was a vasodilator. Now you know the mechanism of how these two work. They work specifically on the kidney level. So that completes the loop of these hormones. And then we'll talk more about the RAS system in more detail in terms of the tubule. All we've talked about so far, keep in mind, all we've talked about so far is the glomerulus for the purpose of filtration. So now that we have an idea of what's happening in the glomerulus for the purposes of filtration, GFR is one of the values that we're looking at in terms of kidney function. And the normal GFR for men is about 125 milliliters per minute in males. In females, is about 150 milliliters per minute. This is normal. 105. Did I say 150? Yeah. 105. My numbers are starting to look a little mixed up, especially at 9 o'clock at night. Especially towards the, was it 14th week now? 14th week? Yeah. 125 and 105. Don't trust what I say, trust what I say right on the board. 105 milliliters per minute in females. This is how much blood, filtrate is formed in the glomerular capillaries. We don't want an excess of that because that's going to cause a lot of filtrate forming through, and they could end up destroying some of that, the pedicels and the sensitive filtration system that we have in place. We don't want too little because then that's not good kidney filtration that's taking place. So our way of being able to measure normal values, we can measure, we can estimate based on urinalysis. And typically they would look at creatinine values. They look at serum creatinine and then creatinine in the urine. And they can estimate the glomerular filtration rate. And anything below this is indicative of kidney disease. And we see different stages at which we see decreased glomerular filtration rate. Let's start with the most extreme version, kidney failure. If you have a very low GFR, that means no filtration, very limited filtration is taking place in the kidneys. If your kidneys can't filter, you'll have to rely on an external dialysis machine to do the filtering for you. And as we start to move in lesser amounts of decrease in the glomerular filtration rate, you can see the different stages. So anything greater than 90, but lower than our normative values, will indicate stage 1 kidney disease. Okay, so you have slight kidney damage with a normal glomerular filtration rate. So typically they won't relate it kidney damage due to the glomerular filtration rate. They're looking at some abnormal urinalysis of some kind, something else that's indicating kidney disease. Any more decreases in the GFR relate to stage 2, 3, 4, and then 5 of kidney disease. And again, that's the dysfunction of the kidney in terms of filtration.
Sometimes it says
End-stage renal failure is basically stage 5. So that point, your kidneys are pretty much on the last legs. They're failing, and you're probably put on dialysis. You would need a kidney transplant at that point. That's pretty much it. So far, all we've talked about is filtration. Again, all this is related to filtration, but going back to, we need to talk about reabsorption, and we need to talk about secretion. Again, reabsorption is stuff, all we've talked about is the fluid flowing through the glomerular capsule, out into the tubule. We're going to need to reabsorb that stuff back in. All the good stuff that was present in the filtrate, our glucose, our ions, our water, needs to get reabsorbed back into the blood. All the stuff that we need to get rid of afterwards, that's present in the blood that we didn't get rid of in the first place, is tubular secretion. That's stuff that's going back into the urine, and stuff that we'll talk about on Thursday. All right? So that's it for today. I will see you guys on Thursday. Have a good night.